Okay, so it's my pleasure to introduce today's speaker. Today's speaker is Carson Nye. Carson grew up in Michigan and got his bachelor's degree at Michigan State. And although he was majoring in, let's see, genomics and molecular genetics, he did actually spend time working in the viticulture lab. He applied to the master's program and started here just after he finished at Michigan State and joined my lab about a year and a half ago now, I suppose. Um, it is always an interesting time right now because we've only met in person a very limited number of times. Mostly we've been working at like this, but I will say that Carson has done a terrific job of being flexible as his project changed in the middle of COVID, and working along with all the things that came up to come up with a very interesting thesis, working on ethanol tolerance and Saccharomyces cerevisiae. So it's been a pleasure to work with him, and I'll turn it over to you, Carson.
Great. Thanks, Dr. Block. Um, like you said, my name is Carson Nye, and my project was evaluating genes of interest for their effect on ethanol tolerance in Saccharomyces cerevisiae. So different genes that, for whatever reason, in the literature I picked out to either increase or decrease ethanol tolerance in hopefully a predictable way. So kind of the motivations for the project, as I'm sure everybody is aware, stuck and sluggish fermentations are a problem in the wine industry and alcohol production as a whole. Sluggish fermentation, something that's running unusually slow, or a stuck fermentation that ultimately doesn't finish. And these are caused directly by an inability for the yeast to continue importing the hexose sugars that it feeds on to produce ethanol. And this can be for a variety of reasons: temperature changes, nutrient deficiencies. But for the purpose of this project, we're going to focus on the rising concentrations of ethanol in the environment. So sluggish fermentations can tie up tanks much longer than expected and cause logistical problems. Stuck fermentations can cause stylistic issues where you don't really want any residual sugar, but you have some. This can leave it open to microbial spoilage. And stuck fermentations, it can be very difficult to restart if you're just going to add more yeast into your must, then the same problems will still be there, but this time with higher initial alcohol. And as I'm sure we remember from our redox presentations in this course, typically by the time you see a stuck fermentation coming in your Brix curve, it's probably unavoidable. So ethanol toxicity in stuck fermentations. The yeast creates ethanol, and that's the reason we use it, but this also creates an increasingly hostile environment for the yeast. It's doing this to essentially eliminate different sorts of competition and to generate energy. But it will eventually succumb to that. The ethanol is going to disrupt your membrane protein function and inhibit your ability to continue taking in your sugars, and is eventually going to lead to cell death. So when you think about it, you can do something, temperature control, to control your temperature and prevent these spikes that will cause your stuck fermentation. You can alter your nutrients, but at a certain point, you can't really do anything about the ethanol tolerance. There's an upper limit there to the amount of sugar that your yeast are capable of fermenting. And the idea here is essentially to hopefully increase that. So building off of previous research that people have done on a variety of different genes and different pathways, we picked some to see if we could differentially express them in mutants and to determine how maybe those differential levels of expression are going to impact the ability of yeast to continue fermenting in the face of large ethanol concentrations. And before we get much further, it's important to kind of define ethanol tolerance and the way it's measured. Often when people do experiments involving ethanol tolerance, they just kind of expose an inoculation of yeast to concentrations of ethanol in what's called ethanol shock, and then they'll measure the viability. That, or they'll grow mutants on like a YPD media that has already been supplemented with ethanol. And this isn't really analogous to a commercial fermentation for a couple of reasons. First, just because a cell is viable doesn't mean that it continues fermenting, and just because if it's not continuing to ferment, it's not particularly useful. And then secondly, when you're growing something on a media that already contains ethanol, you're subjecting that yeast to the ethanol stress during its growth phase. Whereas if you're running a conventional commercial fermentation, there really isn't a sizable concentration of ethanol until that yeast has already entered stationary phase of its growth. It's a much more mature inoculum. So what does ethanol actually do to the yeast cell? How is it killing it? Why is it bad? And the primary target is the cell membrane. You'll see on the left is a pretty typical drawing of a cell membrane. You have your phospholipids, some proteins, your sterols, and that's without any sort of stress. And then over on the right, we see those little dots are ethanol molecules, and they've stressed out the membrane. And you can see how the phospholipids are kind of pushed apart, the fatty acid tails are mangled, the proteins are all pushed around. And this, as we'll discuss in a second, is a result of the ethanol's interactions with the phospholipids. And it's going to cause an increase in membrane fluidity, and it's going to let more passive proton flux into the cell, which will eventually cause your depolarization and end up messing with your redox balance. So extremely high concentrations of ethanol cause what's called interdigitation, where on the left you'll see a standard unstressed membrane. You have your sterols there in yellow, and then your phospholipids. And on the right is an extreme example of full interdigitation, where the ethanol molecules have actually binded to the glycerol moiety that connects the fatty acid tails to your phospholipid head. And then the methyl group plunges down into the membrane to try and hide itself, and the hydroxyl group reaches out towards the polar solvent. And what that's doing is it's kind of wedging the phospholipids apart, and it's pushing them further and further away from each other, which is what's going to cause your increase in membrane fluidity and your increase in passive proton flux. And eventually, these fatty acid tails get far enough apart that they kind of slide in on each other like a zipper. And that's going to cause a really stark reduction in the thickness of your membrane, which will be important in just a moment. So this interdigitation causes this huge reduction in the membrane thickness, and that's going to expose different portions of proteins and the phospholipids themselves that are meant to be hydrophobic and are meant to be dissolved in the membrane that now are exposed to your polar solvent. And this is called hydrophobic mismatch. So now your polar solvent is essentially attacking your hydrophobic portions, and what the proteins and these phospholipids are going to do is they're going to aggregate to try and limit that sort of contact. And this is probably going to cause your proteins to be non-functional, and it's going to tear up your membrane. And even if this doesn't cause it directly to be non-functional, there are non-annular lipids and there are annular lipids. Your annular lipids kind of hold your protein in place, but non-annular lipids act as cofactors with these membrane proteins. And if your membrane is all pushed around and this non-annular lipid gets away from where it's supposed to be acting as a cofactor, then you're again going to see non-function in your protein. And this is what's going to cause serious damage to your cell here. So the big thing is maintaining membrane thickness. Well, how do we do that? And the idea basically is in this ratio of your unsaturated to your saturated fatty acids within your bilayer. You'll see on the left is that extreme example of full interdigitation. And on the right here is supposed to be unsaturated fatty acids that have a double bond someplace, and that causes your fatty acid to kind of splay out a little bit. And the idea here is that the glycerol moiety is still going to be bound by the ethanol. It's still going to be pushing these phospholipids apart, but because the fatty acids sort of splay out a little bit wider, it's going to be much more difficult for the membrane to interdigitate. The phospholipids are going to have to be pushed further apart before it can slide past the other fatty acid. And hopefully this is going to maintain the membrane thickness, and you're not going to be subject to those issues with the hydrophobic mismatch. So the fatty acids in the yeast membrane, they're mainly composed of carbons 14, 16, and 18 chains, and there are only monounsaturated fatty acids. The yeast cannot synthesize any polyunsaturated fatty acids. We have two examples here. There's the palmitic, which is the saturated C16, and oleic acid, which is your C18, with one unsaturation at between the 9th and 10th carbon molecules. So that brings us to the first gene that I looked at, is the fatty acid desaturase. There's only one of them in yeast, And if we want more of these unsaturated fatty acids in the membrane, it would make sense that we probably want more of this fatty acid desaturase activity. What it does is it catalyzes a reaction that puts a double bond between carbons 9 and 10 to make this palmitoleic, the C16-1, and the oleic acid, the C18-1. And it does require oxygen to do this. So I'm not the first person that has thought to use this gene. It's kind of an obvious one, you would think, if this was the tack you were sort of taking. And this here comes from an experiment from 2011 where they took a laboratory strain of Saccharomyces and they overexpressed OLE1, the typical that you would find in your Saccharomyces, but then they overexpressed also these two below the highlighted one, which will make polyunsaturated fatty acids. And what's interesting here is when they grew it up on the YPD plus the ethanol, the oleic acid that's producing these monounsaturated fatty acids is able to survive at a lower cereal dilution in the 10% ethanol and grow in the face of that. But the polyunsaturated fatty acids are not, suggesting that we want, these monounsaturated fatty acids and that maybe they are splayed out a bit more. So here's an experiment where somebody deleted OLE1 and then chose to supplement the media with different fatty acids that they used. And you'll see this figure on the right. There's some pretty clear differences between the fatty acids that they used. And essentially the takeaway here is that this delta 9 C18-1, that's the oleic acid that you're going to find occurring in your yeast from this OLE1. And that is the winner, that had the highest biomass at the end of the 48 hours, growing in the face of a small to moderate ethanol concentration. And it appears that where the saturation is also matters, because you look at that C18 unsaturated fatty acids, the open circle, the open triangle, and the open square. And you'll see that the difference between those is where the point of unsaturation is. And this is a little bit further down the chain. So we see that we really want this delta 9 C18-1, which is what's produced by the OLE1 gene, and increasing concentrations of the C16-1, which is also produced by OLE1, but from a shorter fatty acid chain, is somewhat less effective for the yeast in the face of ethanol. So basic fatty acid synthesis up to OLE1, which is making our unsaturated fatty acids. We start with our acetyl-CoA, and then that is carboxylated by this acetyl-CoA carboxylase produced by ACC1. This is the rate-limiting step in de novo synthesis of fatty acids in the yeast cell. That turns it to malonyl-CoA, which is then taken by this FAS complex, which is a 2-complex protein coded for by the FAS1 and FAS2 genes. They'll attach 2 malonyl-CoAs together and then continue elongating it by adding acetyl-CoAs to the end. And they can do this mostly to the 14, 16, and 18 carbon chains, but they do go a bit further sometimes and a bit shorter. There's these ELO genes here, which is actually a collection of genes that code for different elongases, which have a somewhat overlapping function with FAS. There's quite a few of them. They will only elongate something that's already at least 14 carbons long. But interestingly, they will actually elongate something that's already been desaturated. Whereas once OLE1 gets at the saturated fatty acid chain, FAS will no longer continue to elongate it. So ACC1 is the next gene that we're going to look at that I picked. And this is coming from an experiment where they phosphorylated part of the DNA to get it to be hyperactive. And you'll see the hyperactivity of ACC1 caused a pretty significant rise in the C18-1, that oleic acid that we want, and a big rise in that C18 to C16 ratio. So the idea is that if we get more of this C18 and kind of the same level of desaturation, then we should end up with more C18-1 over the C16-1, which is ultimately what we want. And the same sort of trend was observed back in the 80s. Instead of hyperactivity of ACC1, it was just supplementation with the product that ACC1 makes, this malonyl-CoA. And then the FAS complex kind of goes the other way. So the FAS complex is what's going to elongate after you have your malonyl-CoA from the ACC1. It's, like I said, two complex protein code by FAS1 and FAS2. There is some conflicting information in the current literature about whether or not if you overexpress one, you will also get more of the other. Some studies look at it and say, hey, we overexpressed FAS1 and we saw, just an increase in the total FAS complex expression. And some don't see that. Here we have an experiment that did not see that. This was done in Japan with sake yeasts, and they were looking at overexpression of the FAS complex and its effect on medium chain fatty acids, so shorter ones, things that we don't want for our ethanol tolerance. And you can see the FAS2 gene didn't really have a difference between the control, it's just the vector. The FAS1 had a pretty considerable and observable difference, but when overexpressed together, they saw far and away the most of the FAS complex. And then in some unpublished data from the Block lab of a previous student, observed pretty similar reductions in average fatty acid chain length, and you saw slower fermentation, reduced biomass, just overexpressing the FAS1 gene. And then moving away from the membrane quickly for our last two genes is towards redox. And increased permeability in the membrane, more of these protons are coming in, and increased NADH usage to produce these fatty acids, along with reactive oxygen species generation, are going to make it harder for the cell to maintain its redox balance. And kind of the two weapons that it has to fight back that I decided to look at were glycerol production and efflux, and then these superoxide dismutase enzymes that are going to sequester the reactive oxygen species. So FPS1 is the gene that I picked for the glycerol efflux. It codes for a membrane protein that is a glycerol channel. And it has something of a regulatory role in how much glycerol is actually produced by the cell. So this comes from a big genomic experiment where they screened a whole bunch of different genes to find what they thought were kind of the most interesting with regard to ethanol tolerance in particular. In the graph we see here on the left, there are three lines all right on top of each other and then one down below. And the three on top of each other all have FPS1 expression to some degree. They either have it overexpressed, naturally occurring, or deleted and put in on a vector. And then down below is just an FPS null mutant that didn't reach sort of the same biomass and didn't reach its peak biomass near as quickly. And then in that middle graph is a graph of the ratio between intracellular and extracellular radiolabeled ethanol. And basically what we're seeing there is that increased expression of FPS1 showed smaller intra to extracellular ratios. So the idea is essentially that maybe this glycerol channel is also pumping or allowing ethanol to move out of the cell. And then over on the right is a fermentation curve of an overexpression and a typical control. And we see that the FPS1 overexpression produced higher levels of ethanol and utilized more sugar. So the fermentation essentially went further, which is exactly what we want. And then our last gene here is this SOD2 superoxide dismutase. This is a mitochondrial superoxide dismutase. It has a cytosolic brother, I guess, this SOD1, which is interestingly enough a gene that mutations in it can cause ALS. So there's a decent amount of research done. And the Sakasa et al. In 1997 looked at its levels of expression in yeast in the face of ethanol at different points in its life. And it saw that both SOD1 and SOD2 increased in the expression up through exponential phase, but once it reached stationary phase, remember where we're getting the bulk of our ethanol, SOD1 kind of leveled off and SOD2 continued increasing in its activity. And then mutants deficient in SOD2 showed lower viability with ethanol shock. And then Lee et al. Here took this SOD2 gene, which of course isn't normally found in E. coli, and they put it on a vector in some ethanolic E. coli and found that the mutants that expressed this had a greater viability when exposed to low concentrations of ethanol. So that was a lot of genes, and here's a quick summary of the ones that I picked. We have OLE1, our only fatty acid desaturase in yeast. We have ACC1, the rate limiting step in our fatty acid synthesis. We have FAS1 and FAS2, which make up the two complexes in the fatty acid synthase. And then we have FPS1, our membrane channel for glycerol efflux, and then SOD2, our superoxide dismutase. So what did we do with those? Well, Rachel Monpetit made a variety of different mutants that are single gene overexpressions in 2 different commercial wine strains. So that would be 12 mutants and 2 controls for 14 in triplicate, which I ran in 2 fermentations, 1 for each strain, which I will talk about in just a moment. These I inoculated into a Hybrix triple M media. This is a minimal must media. It's designed to sort of emulate a grape must, but I put enough sugar in it to bring it up to 28 Brix, which is certainly not the standard. And the idea here was essentially to put enough sugar in there that the fermentation would stick, and we could see kind of where they stick and use that as a good metric for how ethanol tolerant something was. These were fermented in a temperature controlled shaker in 400 milliliter flasks with stoppers on them. And I monitored Brix with a DMA, OD600, which is our optical density, our biomass, nitrogen utilization, and then dry cell weight, which is essentially kind of measuring the same thing as the OD600. It's just the dry cells that I've filtered through and then dried out the filter and weighed. And then the most interesting of these we would later take for HPLC analysis for the sugars, glycerol, and ethanol of just our most interesting mutants. So the 2 strains that we picked were picked for a reason. They were picked because they were quite different in terms of their ethanol tolerance. You'll see the orange there is Elixir. It ferments down further, it ferments quicker, it develops more biomass there on the right, as opposed to Montrachet, which doesn't ferment quite so far and doesn't ever reach the same sort of biomass as Elixir. So our initial screening in this Hybrix with the single mutant overexpressions. Here's the Montrachet, and we see not crazy differences between most of them, are about the same, but we see this FPS1 gene stopped fermenting about a Brick earlier, which is not what we would expect based on that Texera et al. 2009 data, but it's something, certainly. And we'll see the SOD2 gene here had a bit longer of a lag phase and it was a little slower to get going, but it ultimately fermented to the same level as the others. And we see that reflected here in our OD600 data, where this is the optical density, and we'll see that FPS1 never really reaches the same sort of cell mass as the others do, which our dry cell weight agrees, which is good. We kind of see that slower rise in SOD2. Everything else is about the same, and then the FPS never really reaches the same as the rest. And then we get to our yeast assimilable nitrogen. We see that most of them are about the same, but as the cells begin to lyse towards the end of fermentation, where there's a lot of ethanol there, it looks like FPS1 may be lysing a little bit quicker. It's kind of giving that nitrogen back into our medium. And then how did Elixir do? The same sort of data. Here's our initial Brix curve, and we see this time FPS is a little bit higher maybe, and FAS is up there with it, and everything else is kind of in a cluster just slightly below. For the OD600, the optical density, it looks pretty similar, but I cleaned it up just a little bit to show you this small difference here. So there is a difference in biomass, but it's very slight. But both the FAS1 and the FPS1 that seemed to stop a little bit earlier also produced slightly less of a biomass. But the dry cell weight doesn't necessarily agree with that. But the dry cell weight has more variability to it. It's kind of a more complicated and involved way of measuring this. Just my own anecdotal experience in the lab. And then there was really no difference in nitrogen utilization at all between any of the strains or of mutants for this strain. Great. So what did we learn from our initial fermentation there? The Montrachet FPS1 mutant stuck before the control. The FAS1 and FPS1 may have stuck a little bit before the control in the elixir, but certainly led to reduced biomass formation. So where we went from here was we decided to try FPS1 deletions instead of an overexpression, and then an overexpression of the full FAS complex, so both FAS1 and FAS2, to try and see if we can exaggerate the differences that we saw. So Rachel went back to the lab and made those mutants for me. Thank you again. And while she was doing that, I tried to replicate our results, the FPS1 overexpressions, but in a lower Brix media. You know, FPS1 was the most different, certainly, of all the genes. So we wanted to see if that would hold true if it weren't in the face of high concentrations of sugar or ethanol. So that's what I have here. And we don't see really a huge difference. The FPS1 mutant may ferment a little bit quicker towards the middle of fermentation, but of course this is a lower Brix media and they will all finish, and so they do at the same point. We definitely don't see that huge reduction in cell mass that we were seeing at the high sugar concentrations. There is a reduction kind of towards the end of the fermentation, but it's certainly not that green line we saw earlier that was significantly lower quite quickly from the control and really the rest. And we see a little bit here at the end for the elixir, but not, again, as much as the Montrachet. And our dry cell weight, even though it is only 3 time points, does seem to agree with that. We see towards the end of fermentation a pretty significant difference in the biomass between Montrachet for the control and the FPS1 mutant. In this case, there wasn't really a difference in the nitrogen utilization between either elixir or Montrachet. Remember, Montrachet kind of lysed quicker and gave up that nitrogen back to the media. It looks like it might do that a little bit right at the very end of there, but certainly nothing like it did in the high-Brix media. So what we learned from that was essentially, again, Montrachet was affected more than elixir by this FPS1 overexpression, but it was a much more muted effect at the lower Brix than it was at the higher Brix. And it was like immediately muted if you look at the biomass data. So at this point, the cavalry has arrived and my FPS1 deletions have been prepared, the FAS1 overexpression, and so we ran those as well in another high-Brix media. And this is where things I think get really exciting, because you can see here in our Brix curve, one thing is not like the other. And that is our Montrachet control sticks before everything else. But our Montrachet null mutant for the FPS1 made it about 2 Brix further than the control. So we're seeing it essentially continue fermentation further in the face of this rising ethanol concentration. What we don't see is really any difference with the elixir. It's very small if it's there, but certainly nothing as exaggerated as this Montrachet. We see kind of a similar thing with our OD600, the biomass data. We see a pretty significant increase in the biomass for the FPS1 mutant as compared to the control. That's the blue to the gray there. And we see a small one for the elixir, but certainly nothing to the extent that Montrachet shows. And again, our dry cell weight data agrees with that. We have our control at the bottom and the elixirs and the Montrachet null FPS, a good deal about that. And then the nitrogen utilization. For the null FPS1 behaved a lot like the elixirs, pretty much identical to them. Whereas our Montrachet control is as it was before. It uses the nitrogen a little bit slower, doesn't use as much, and then tends to give it back a little bit more quickly. So our FAS complex, this we see kind of the opposite, where Montrachet doesn't seem to really be affected at all by this full FAS complex overexpression. But we see that elixir ferments slower and not quite to the extent that the control does. And we see this again with the OD600 data, where the elixir control has much more biomass than the full FAS complex overexpression. The full overexpression causes a pretty significant reduction pretty quickly in the biomass. And then a small difference for Montrachet, but not nearly so as with the elixir. Our dry cell weight agrees with that, but I mean, again, you can see the variability there. our error bars. Here, the YAN wasn't particularly different between the controls or the FAS mutants, but we see maybe a slight difference there with the Montrachet. So our level of FPS1 expression affects our Brix and our biomass in both strains, kind of in both directions, but with Montrachet far more than it affects Elixir. And kind of the opposite direction is the FAS complex seems to really affect the Elixir, but not really the Montrachet too terribly much with our Brix and biomass. So these are kind of our interesting genes that we used. And so these are the ones that we kind of looked at when we were doing our sugars, glycerol, and ethanol analysis. And you'll see here our glucose from that initial high Brix, that initial screening of kind of our more interesting genes. I had a dilution error, I think, with two of the samples, one in an FAS1, kind of the last replicate there, and the first replicate of the control. And so that would be why the glucose seems to go up there. But I think the really interesting thing, other than my shame there, to see about this is that the sugar isn't really different. But we saw in the Brix curve from the initial that the FPS1 stopped earlier for the Montrachet. And we don't really see the fructose compensating for that at all. And I think the reason that this could be is because the DMA is a densitometer, it's measuring relative densities. And I think that maybe the massive increase in glycerol that we're seeing, glycerol is very dense, could be kind of skewing that data and making it appear as though the FPS1 is not fermenting quite as far as the control, when in actuality, it's using the same amount of sugar, it's just producing more glycerol. And we see here, kind of in line with this idea that the FPS1 really affects the Montrachet and not so much the Elixir. There's a huge gain in glycerol production and efflux with the Montrachet, and a gain, but not quite as exaggerated here with our Elixir. And then the ethanol, Montrachet produced a final ethanol concentration of slightly less than our control, our FAS, which makes sense. Ethanol and glycerol are both made of carbon. They're kind of competing products here. So if we're going to see more ethanol or more glycerol, we would expect to see a little less ethanol. And then we're not really seeing a huge difference with the Elixir. And with these FAS1 genes, we're not really seeing a huge difference either way, which is to be expected because this isn't the full complex. That comes later. And for the interest of time, I only pulled out the glycerol graph from the low Brix FPS1 fermentations. Nothing was particularly different between control and treatment in these particular fermentations. Except for this glycerol here, you'll see again, just kind of this idea of the blue and the gray are the Montrachet, and we're seeing a lot more glycerol with the Montrachet FPS1 overexpression. There's a much bigger gain there than with the Elixir. So our FAS complex, the whole complex put together that we've overexpressed here, we're seeing now kind of the opposite of what we saw with the Montrachet and the FPS1. We have one of these things is not like the other, but this time the Elixir control has gone down much further than the Montrachets and the Elixir with the FAS. So the FAS complex overexpression didn't really affect the Montrachet too terribly much in terms of sugar utilization, but it did cause the Elixir to ferment more slowly and not quite to the extent that the control did. And with ethanol production, there was not a huge difference. Again, no difference between the Montrachets, but the Elixir ferments perhaps a bit slower, but ultimately reaches about the same concentration. And then our null FPS, this agrees with our initial Brix estimates this time. We see our Montrachet control is a little higher. Our Montrachet FPS1 has now fermented lower than the control. Really nothing between the Elixirs. Glycerol production, as we would expect, since there are no longer these transmembrane proteins to export them. Glycerol for both nulls is about identical and lower than the controls. And then our ethanol here is kind of the really exciting graph at the end. We see that our Montrachet control and our Montrachet null FPS are not quite the same. The null FPS ended up producing about 1.6% alcohol by volume more than the Montrachet control. So essentially, it increased the ethanol tolerance. It was able to ferment further in the face of this rising concentration of ethanol. And interestingly, we really didn't see that with the elixir. So it appears to be kind of strain dependent. This FAS complex, this increase, assumably in the medium chain fatty acids, seemed to have more of a deleterious effect on the elixir, whereas the getting rid of these membrane proteins seemed to benefit the Montrachet much more than the elixir. Great. And just a quick word of thanks, I suppose, to my family, of course, my biggest supporters, to Dr. Block for letting me play mad scientist in his lab for the past year and a half, Rachel Monpetit for the hours and hours of making mutants for me. I would still be scratching my head at a lab bench without her. To Ben Monpetit for his advice in picking genes and help with the data. Ying Choi for helping me use the HPLC. It's harder than it looks. And then, of course, to my classmates and my financial support here. Do we have any questions?
Great, Carson. Any questions for Carson? I have a question for you. Maybe get us started. So, so one of these genes you, I think, overexpressed and knocked out.
Yeah, that'd be the FPS1 gene.
So, so were the results consistent there when you went from, let's say, a null to wild type to an overexpression?
Not really. That's kind of what I was saying with the increased glycerol production and the increased density there, because we're seeing with the overexpression of FPS1, the DMA shows that the density has stopped, before the control. So it looks like the fermentation hasn't lasted quite as long. But when we got to the HPLC and we looked at the actual glucose and fructose measurements still in there, they were not different, but the glycerol was significantly different. But when we compare it to the null mutant, we actually do see it ferment further. It utilizes more sugar and it produces more ethanol.
Cool. Other questions for Carson? Oh, well, people are thinking questions. I'll ask another one. So, given what you know now about these genes, if you were thinking about using this to come up with a new strain, what would you think about doing? What kind of strain would you make?
I think the first thing that I would do is I would do this again, but I would add dissolved oxygen. Because like I was saying at the beginning, that OLE1 gene that's producing our oleic acid that we really want, it requires oxygen to do that. And while this was an anaerobic fermentation, I wasn't taking super care to keep oxygen out, but I also certainly wasn't dissolving oxygen in there. And I think that could be a big reason why we really didn't see a difference with our OLE1 gene, the ACC1 gene, just because I don't think, you can have as many longer chain fatty acids as you want, but if there's not enough oxygen to turn those into your oleic acid, then you're not going to get your oleic acid. So I think that would probably be the first thing that I would do, and then I would kind of go from there. But based on just the results that we have here, I would say that if you wanted to make a more ethanol tolerant strain, a pretty good way to start would be by deleting FPS1 and at least seeing what would happen based on your base strain, because we saw, some strain differences there. The Montrachet was quite affected, but the elixir wasn't, which tells me that there's probably going to be some variability there between other strains as well. But I think it would be a pretty good place to start.
Do you think you can have too much oleic acid in a yeast? I don't know the answer to that. I'm just wondering what would happen if you had too much.
I don't know. I suppose that would be probably a pretty easy thing to find out. We could just add a whole bunch of oleic acid and see how that affects the growth. Um, but I would imagine that at a certain point, it's probably going to hurt the yeast, because if the yeast were going to be better, if every single fatty acid was oleic acid, it probably would have done that itself a long time ago. I mean, why would it be producing palmitic acid, palmitoleic acid, if it didn't need it for some reason?
Want to, you didn't talk very much about the head groups on the lipids, but I think there's in the literature from our group and other groups and so forth, there's a seems to be some impact of the head group, and so I wonder if like oleic acid attached to some head groups will
Mm,
actually make a difference, and oleic acid attached to other head groups maybe less so. I don't know, and so there's maybe some limit in concentration where adding more oleic acid will work, but I don't know that for certain.
yeah, that's for future master's students like me.
Yep. Well, I could go on talking about yeast physiology forever, but I'm going to let anybody else ask a question if they'd like, because I can always chat with Carson later. Any other questions for Carson?
I don't have a question, but I don't mind listening to you chat. I'm learning a lot in this portion as well.
Thanks, Andrew.
Yeah, I learned more than I ever thought there was to know about the differences in fatty acid chains in yeast, and fatty acid synthesis and all that. So this was certainly a learning experience for me.
Well, and I have to say this is part of a larger effort in our lab to try and we did some work with Dario's lab and Ben's lab a while back on looking at kind of all the omics of several of these strains where they weren't genetically modified to understand the differences. And more recently, we've come up with some kind of genome scale metabolic models, mathematical models for what's going on. And the idea was originally was that um, the data that Carson generated would be data that we could use for the modeling efforts to, once Carson figured out what was going on with these overexpressions or knockouts, that we could actually simulate that from in the mathematical modeling and see how good the models were. Because if the models are really good, then we can. It certainly takes a lot less time to simulate something rather than doing weeks or months or years of experiments. And for various reasons, related to the pandemic and other stuff, Carson got all his data before we were able to do anything with the modeling in this area. So my hope is that we'll still be able to take these data, and see and test the model with it eventually, hopefully in the next year or so. All right. I think if there are no other questions, I think that let's thank Carson again for a great talk. And yeah, congratulations on being nearly finished.
Thanks.
Well done, Carson. Congratulations. Good. Thank you, everyone. Thank you, Dave. And see you later. Bye.